All right, well, welcome everybody. We're really excited for you all to be joining us today. We are coming from the California Academy of Sciences, and my name is Jackie, and I have my friend Lindsay with me.
Good morning, everyone. I'm so excited to have you all join us today.
And we got to hear a little bit about what you all think about what is spookiest, whether it's bugs, guts, or skulls. And Lindsay, did we get a chance to actually see what people said?
We did. So here is what popped up for what people think is the most spooky. And I think it's really interesting to see. It looks like guts is something that's kind of winning out for that.
Yeah, and then also we asked what would be the most interesting to study.
Yeah, so let's see if it's similar or different.
Well, there we go. Interesting. Skulls is seems the most interesting to study.
Yeah, bones in general, which is kind of great. I have my bone outfit on today.
Yeah, I do too. I got my skeleton on. Because we're going to be diving in a little bit deeper into skulls and bones and guts. And we are actually kicking off our first session in our 3-week series today, which I'm really excited about.
Absolutely. So we're going to be meeting for 3 weeks in a row in this place at this time to explore these different topics: bugs, bones, and guts.
Now, today we're going to dive a little bit deeper into bugs, but also into bones. So we're going to kind of dive into both of them. And we also asked you to kind of think about one room here at the Academy, and we'll be able to dive into that a little bit later. Oh, looks like we're actually going to get to see some of those ideas coming up.
So yeah, I noticed a few different things. It looks like bones is kind of in the middle. Maybe it's used for bones or storage or painting.
Yeah.
Well, like Jackie said, we're going to dive into that a little bit more, but we wanted to just take a moment and introduce ourselves to you and where we're coming from. So we're coming to you from the California Academy of Sciences. It's a museum in San Francisco. And one of the things that we love to do is we love to explore nature all around the world. So we have a giant team of coworkers and scientists who all are linked together.
Yeah, and one really cool thing about the Academy of Sciences is I have wonderful coworkers like you, Lindsay, but we also have some other coworkers that are some of my favorite. Our living coworkers, some of the butterflies and penguins, and even Claude, our albino alligator.
Yeah, and so we like to have them with us so we can care for them, keep them healthy. And in addition to that, they help us learn about nature and plants and animals and the world around us so we can care for plants and animals outside of our museum.
Now, some of our other coworkers aren't actually living. They're what's behind me and also behind you, Lindsay. Those are our non-living collections.
Yeah, so, it's interesting. I know a lot of our friends and scientists who are logging on today have had an opportunity to go to a museum, and sometimes you'll see what you see behind me, like in an exhibit or something, but there's areas that are a little bit different. And so I thought we'd hear from one of our other coworkers, Mo, just a little bit about her work and what she does, because she takes care of our non-living collections.
Hi there, I'm Mo Flannery. I'm the collections manager in the Department of Ornithology and Mammalogy here at the California Academy of Sciences. I'm actually something like a librarian, but instead of taking care of books, I take care of specimens. In ornithology and mammalogy here at CAS, we have over 100,000 bird specimens, 30,000 mammal specimens, and 11,000 eggs and nests. And researchers from all over the world can come to the collection to use those specimens, just like you would go to a library to read a book.
Wow, I never really thought of a museum as a library.
Yeah, it's one of the interesting things that happens. So only a little small bit of what we see in museums is what exists in a museum. And in the basement or in different labs, there's a bunch of other things to explore. And that's what scientists do. So one of the main purposes of natural history museums and aquariums is to do science.
All right, well, Mo was talking about things coming into the museum. And so I'm kind of curious about this question of how do those bones and those specimens get into our museum? And I think we're going to kind of explore that today.
That's exactly what we're going to explore today. So let's just kind of take a moment and to think about this for ourselves. How do bones get into a museum? Just think for yourself for a moment how that happens. And what we're going to do is we're going to follow the path of a particular set of bones and things that have been brought to our museum today.
Now, I know we get our information from a lot of places around the world. We are connected with people that love to explore nature and do science. And this map represents just some of the many places that we get our information from.
Yeah, and what's really interesting is this map is mostly made up of people around the world. Everywhere you see orange or yellow or red, that's where we've gotten information sent to us. Sometimes people send us pictures of things. Sometimes they send us actual real things to look at. And so it's really interesting, but there's this whole network that we all work together to learn about. So I have an interesting story for you.
Okay.
So down near San Luis Obispo, which is south of San Francisco, it's on the California coast, there were some hikers recently who were going along this place. It's called Villa Creek. And when they got to the bottom of the cliffs, they found something really interesting. At first, they just saw all sorts of birds kind of like flying around. And when they got closer, they started to smell something and they started to get a little bit closer. And what they saw was something like in this picture.
So, what did they do when they found this object?
Well, they kind of, walked up to it and they were looking at it closely and they thought, this is really interesting. We don't really know what it is. And so they contacted the California Academy of Sciences, and in particularly Mo, to say like, hey, we found this thing. Is this something that you might be interested in? And so she was interested in it. So we had one of our vehicles go out and bring that object back, that kind of, it's called a carcass, it's a dead animal, bring it back. And instead of bringing it through the front, where you pay with a ticket to get in or you have your free pass to get in, we bring it to the back to an area called the loading dock and into this place called the Ornithology and Mammalogy Prep Lab. Now, for some of our scientist friends who saw a picture at the beginning, this is the prep lab. It's where we prepare dead animals to come into our museum. And we want to do that really well and carefully because, especially when we have a carcass or a dead animal, that might bring with them different bugs and things that we don't necessarily want in the museum. So we take it to this special room where we can prepare it.
So I'm actually kind of curious to think about this. How should we prepare that carcass? How should we get all the stuff off so we can have nice, clean bones like the ones behind you and I?
That's a really interesting question. So I'm going to throw that back out to all of our friends and scientists who are connecting with us online today. If you can go to the link that's in our box or write on a piece of paper some of your ideas of how might you clean the flesh off the bones of a dead animal. And so we'll give everybody a minute to kind of think about that and just to jot down a couple of ideas. Like, what would you do? All right, so some of the things that I see popping in are these ideas of like, ooh, I might use soap and water to wash it. We might have an opportunity to use some maybe chemicals or to scrape some of the flesh off. Some people say, I don't know.
Yeah. You know, I didn't know exactly how this process happened, but I remember Mo telling me there's a couple of different steps, and I'm really getting confused a little bit on what steps come first and which ones come second. But I do know that we have to whiten the bones. We need them to look nice and clean like this. I do know that we have to get all that flesh off, and we use beetles to do that. I do know that we need to dry it out and make it kind of like jerky, like beef jerky. And then also I remember that we have to freeze it. But those are the 4 stages that I remember. But I can't remember what order they go in.
Yeah, that's a good question. So those are some really good memories and something to really retain. So I want everyone to think for a moment, if you had these 4 steps that we see laid out, which order would you place them in? Would you do D first? Would you do B first? And then what? And then what? So think for yourselves, what kind of maybe might make the most sense to you?
And you can go ahead and go back to that Mentimeter to vote, or you can write down your ideas.
This one is not in Mentimeter.
Oh, sorry.
All right, so let's kind of think about this and let's kind of go a little bit deeper in and we'll see if we can find the answer to this question. So to find this question, we have to go back and find Mo. So we're gonna, again, go back into some of these areas that are behind the scenes and these labs and these collection rooms and really try and figure out where and how do the bones get in there. And to do that, we're gonna be looking in particularly and hearing from Mo again.
So most specimens get into our collection through salvage. And that means that if somebody's walking on the beach, they see a dead marine mammal or they see a dead bird or a dead otter lying on the beach, they'll call us and we go out and we collect data on the specimen. So we collect how long they are, what species it is, whether it's male or female, how old it is. And then often we will collect the skull or sometimes the whole skeleton of the specimen to add to our research collection for scientists to use. And we have to prepare those specimens here in the laboratory at the California Academy of Sciences, which is a really stinky job.
All right. Well, that kind of answers our questions.
Yeah, so it turns out you dry it out like jerky, then you invite bugs to eat the flesh, then you whiten the bones, and then you freeze them so they're all clean and made sure that they're bug free before they go into the collections, because we don't want bugs to get in there and chew up other things.
Now, I would really love to see these bugs at work. But to be able to do that, we need to come back to the academy late at night.
Yeah. All right. So let's see what happens at the academy late at night and why do we need to go there?
So to be able to see these beetles called dermestid beetles, they only do their job when it's dark. So coming to the academy late at night, we can go down into that prep room and watch the beetles work for 4 days straight. So we're going to watch a time-lapse video of the beetles eating the flesh off of this skull.
So think about, what do you see the beetles doing? What's changing about the skull? Jackie, I see some of our friends noticing that the beetles really like the eye to begin with, but now it looks like they move back towards the back of the skull.
Yeah, I'm noticing that too. Like once the flesh is gone, they're kind of go and move on to where there's more flesh. And so this process took 4 days, and this is actually the skull that was found by those hikers.
Wow. So if we look at this kind of before and after picture, I can really see how important those beetles are to that cleaning process. That's really fascinating.
Yeah, so once we have that nice clean skull, we can actually take a closer look at the skull. And Lindsay, I think you happen to have it. And we can use one tool that scientists use to notice details about the skull.
Yeah, I was talking with, this is Rich Moy, he's one of our scientists, and he was mentioning that even though he's been a scientist for a very long time, for over 30 years, he still sketches almost all the things that he finds because it helps him notice details and be able to identify it. And Mo, she knew we were going to be talking with you all today, and so instead of her telling us what it is, she is giving us the challenge of observing a skull and looking at it closely so we can collect details and begin to try and piece together what this animal was, what role it served in its ecosystem. And so we're going to be playing with that just about now. We're going to do that through sketching.
Well, Lindsay, I knew we were going to talk about sketching, so I actually made a sketch for you of a leaf, and I want to see if this is what you mean by scientific sketch. What do you think?
Oh, that's a really nice leaf. I can definitely tell it's a leaf.
Can you tell what type of leaf it is?
That's a good question. Let me bring up some pictures from leaves in our collection. And let's see. You know, I can tell yours is a leaf, but I can't really tell quite the details that might help me figure out which type of leaf it is.
Okay, so for a scientific sketch, it needs to be a lot more detailed. What else does it need to have?
That's a great question. So Rich was telling me that one of the important things about it is to try and make it accurate. So instead of just remembering like what a leaf might look at, you need to spend time looking at it so you can pick out the details and point out where it's really accurate. You also want to draw it big because then you can really hone in on some of the details. And then you can put those in your sketch or you can add these written notes to it. And really that's the art of science. It's really helping to observe and kind of notice these things.
All right, well, I think it would be great for us to kind of look a little bit closer at that skull and do our own scientific sketch. So
Yeah.
for all of you, let's go ahead and first just look at the skull and see what we notice.
So I'm just going to kind of slowly turn it around and we'll kind of start to notice some details. And then that's kind of the first step. So we'll just kind of spend that time noticing what's happening there. And scientist friends, if you can go ahead and get out your pencil and paper, what we're going to be doing next is we're going to be practicing the sketching. And so sketching for science is a little bit of a skill to practice. So we're going to practice that now, and then you'll have an opportunity to do more of it at a different point in time. So let me go ahead and bring up my lab camera. All right, here it is. So, Jackie, I'm gonna move over and I'm gonna put the skull under there so we can kind of look at it closely. And then we'll kind of go through those different steps. And while I step away, my microphone might not catch very well, so just hold back if you can.
All right.
All right, so the first step was to look closely.
All right. Well, Lindsay, your mic is just a little bit down, so I'm going to go ahead and narrate it. So yeah, like you said, our first step is going to be looking closely, see what we notice, and we're going to watch it or look at it from one side. So I think you've picked our side. That's great. Let's start to notice some of the things on our skull. And then next is to start drawing shapes that you see.
So I'm just gonna say I see kind of like a boxy shape and some semicircles. So I'm just gonna start kind of sketching that out. And scientists at home, I'm gonna keep the skull up under the camera. And so you can sketch along and pick out the shapes that you see too.
It looks like Lindsay found kind of like an eye hole, so some circles, maybe some triangles for the teeth.
All right.
So next step is to add some lines, simple lines to this. And it looks like you already started a little bit. I see kind of like this curly Q and kind of some arches. Let's see what other lines we can find. All right. And then our last step is to add details. So we're going to try to add details of what we notice. And we can also use words and also add questions here. So if you notice something interesting or you have a question about this skull, you can write it on your sketch.
Yeah, so I'm noticing like the number of teeth, like on top, there's one big tooth right here, and then these three teeth that are kind of similar. So I'm gonna try and draw that, but I'll also write it.
Oh, and some of our scientist friends are asking if we should put labels on these sketches, and definitely we should put some labels like you are putting right now. That I see 3 teeth. And anything else you notice about this skull? Now, if you're having trouble seeing this skull, don't worry. We'll actually have a picture of this up on our website and after this session. So you can actually go back and look a little bit closer, see if there's any more details you can pick out.
All right, so I'm just gonna kind of pop over here for a moment. And I don't know if you can see it, but one of the things that I'm noticing is that some of the teeth just have like a little bit of a colored part here. So I'm gonna start to add some of those more details on there. And a question I have is why, if this skull was bleached, like why is there still some colors on there? I'm just really curious about that.
Interesting. Yeah, I kind of noticed it's really not really white. And I start to notice kind of at the top of the teeth, It's a little bit different color than maybe at the bottom of the teeth if we look really hard, now that you mention it.
All right, so once again, the sketching, the intent behind it is really just to start to notice some of these details. And there's definitely things I never noticed before. And as Jackie mentioned, we're going to place more of these pictures of the skull so you can see it more in depth on our website.
All right. Well, now that we've kind of done a scientific sketch, I'm kind of curious a little bit more about this mystery skull, and I'm kind of curious what it is. And I think we're going to be able to dive a little bit deeper next time with you all to explore this skull and maybe be able to determine what it is a little bit later.
Yeah, so one of the things that we're going to do is we're going to place in the chat box a link to our student activities page. And within our students activities page, we have some different tips and different things to explore before we go on to our next session. So after our webinar, we have some options for you. So option A is a creative writing activity where you can think about writing either a conversation between a skull and a dermestid beetle, where they each try to persuade each other that they are more spooky. Or maybe you write a travel blog for a skull getting into a museum. Option B is to pick a place outside to look for animals near where you live. You could even just be looking out your window and looking for evidence that they're there. And C is scientific sketching. So we're posting links of pictures of the skull where you can practice more scientific sketching and collecting more details, because we don't have time necessarily today to finish our observations and to figure out what this skull is. So we're going to be working on gathering evidence next time. So doing some more sketches of the skull can help.
All right. Well, we wanted to say thank you, everybody, for joining us today and exploring this mystery skull. And I can't wait to dive in a little bit deeper next time to figure out what this skull is.
And just so we have a little bit of a perspective, what we're going to do is we are going to do a Mentimeter again. And so you can go back to that same one that you did last time, and you can help share some of your thoughts on today, what you enjoyed, what you found surprising, what questions you have. And then that way we're able to have an opportunity to really think about what it is that is happening. So we're going to adjust depending on what it is that we find out you're interested in. And in addition to that, if you have any questions, we're super excited to answer. So thank you so much. I had a blast today, and I can't wait to do more explorations again. See you later. Bye-bye.
Bye, everybody.